Hello friends, today we are going to learn about shuttle system. What is shuttle system or why it is required? We know that in cytosol, glucose is converted to pyruvate. Through glycolysis. And at one step, we get two molecules of NADH. Then this pyruvate enters into the mitochondrial matrix and further oxidation takes place by pyruvate oxidation and Kreb cycle. You can see the glycolysis, pyruvate oxidation and Kreb cycle in detail in my previous videos. This produces more NADH molecule and FADH2. These NADH and FADH2 molecules then transfers their electron to the electron transport chain present on the inner mitochondrial membrane, and then ATP is produced. But what happens to this NADH that is present in the cytosol? It cannot enter the mitochondrial matrix as inner mitochondrial membrane is impermeable to 2-NADH. For this, there are certain shuttle systems present. In this shuttle system, there is carrier molecule present. This carrier molecule then takes the reducing equivalent of NADH. That means it takes the electron and hydrogen ion from NADH. NADH is oxidized to NAD+, which can be used further, and this carrier molecule is reduced. Then this carrier molecule is transported inside the mitochondrial matrix. Inside the mitochondrial matrix, it is converted into the oxidized form and it transfers its reducing equivalent to NAD plus or FAD, which then gets reduced to NADH or FADH2, and this reaction is catalyzed by mitochondrial enzyme. Then this NADH or FADH2 transfers the electron to electron transport chain and ATP is produced. Depending upon whether the reducing equivalent is transferred to NAD plus or FAD, the yield of ATP depends. If it is transferred to NAD plus, then it gives NADH. If it is transferred to FAD, it gives FADH2. NADH gives 2.5 ATP molecule and FADH gives 1.5 ATP molecule. We will learn about two types of shuttle system. One is glycerol 3-phosphate shuttle. This takes place in skeletal muscles and brain cells. Second is malate, aspartate, shuttle. This takes place in kidney cells, heart cells and liver cells. Let us start with glycerol 3-phosphate shuttle. Cytosolic NADH transfers its reducing equivalent to dihydroxy acetone phosphate. NADH is oxidized to NAD plus and dihydroxyacetone phosphate is reduced to glycerol 3-phosphate. This is catalyzed by the enzyme glycerol 3-phosphate dehydrogenase. This is cytosolic enzyme. Now glycerol 3-phosphate transfers its reducing equivalent to FAD. FAD is reduced to FADH2 and glycerol 3-phosphate is reoxidized to dihydroxyacetone phosphate. This FAD is the coenzyme of membrane bound enzyme glycerol 3-phosphate dehydrogenase. It is mitochondrial enzyme present on the outer surface of inner mitochondrial membrane. Then this FADH2 transfers its electron to electron transport chain and ATP is produced. We know that NADH gives 2.5 ATP in electron transport chain and FADH2 gives 1.5 ATP molecules. Here the reducing equivalent of NADH is transferred to FAD. So we get 1.5 ATP molecules instead of 2.5 ATP molecules. So there is loss of 1 ATP molecule. We will learn about malate aspartate shuttle system. So let us start. Malate aspartate shuttle takes place in liver cells, kidney cells and heart cells. It uses antipoters is antiporters. These are transmembrane proteins. These are co-transporters which allow the movement of two or more molecules in the opposite direction. Suppose this is an antiporter. If one molecule moves in this direction, then it is coupled with the movement of the other molecule in this direction. Or in other words, if one molecule is exported, then it is coupled with the import of the other molecule. Let us see this in detail. This is cytosol, this is matrix in a mitochondrial membrane, and it uses two antiporters, malate alpha ketoglutarate antiporter and glutamate aspartate antiporter. In cytosol, by glycolysis, NADH is produced. We know that NADH transfers its electron to electron transport chain and then ATP is produced. But electron transport chain is present in the inner mitochondrial membrane, and inner mitochondrial membrane is impermeable to NADH. So how the electrons from NADH passes to the electron transport chain? In cytosol, oxaloacetate is also present. This takes the reducing equivalent of NADH, and NADH is oxidized to NAD+, which can be used again, and oxaloacetate is reduced to malate. This reaction is catalyzed by malate dehydrogenase enzyme. This malate dehydrogenase enzyme is present in the cytosol, so it is known as the cytosolic malate dehydrogenase enzyme. This malate is then imported by mitochondrial matrix. That means this malate, which is carrying the reducing equivalent of NADH, it is transported to the mitochondrial matrix. In the matrix, NAD+ is present. Malate transfers its reducing equivalent to NAD+, and then it is reduced to NADH. This NADH then transfers electron to electron transport chain and ATP is produced. Malate is then oxidized to oxaloacetate. This reaction is also catalyzed by the malate dehydrogenase. This enzyme is present in the matrix, so it is known as the mitochondrial malate dehydrogenase. So here we see cytosolic malate dehydrogenase and mitochondrial malate dehydrogenase catalyzes the reaction in opposite directions, that is in the cytosol from oxaloacetate to malate and in matrix from malate to oxaloacetate. This depends upon the concentration. In the cytosol, the concentration of oxaloacetate is high, so malate dehydrogenase catalyzes the reaction from oxaloacetate to malate, and in matrix the concentration of malate is high, so malate dehydrogenase catalyzes the reaction from malate to oxaloacetate. Now what happens to this oxaloacetate? This oxaloacetate then takes amino group from glutamate that is also present in the matrix. Glutamate is converted into alpha-ketoglutarate and oxaloacetate is converted into aspartate. This reaction is catalyzed by the mitochondrial aspartate amino transferase. We have seen that a malate is imported by mitochondrial matrix through the antiporter. So this import is coupled with the export of certain molecule. This molecule is alpha-ketoglutarate. The alpha-ketoglutarate is then exported in the cytosol through the malate alpha-ketoglutarate antiporter. Alpha-ketoglutarate. Then the aspartate that is produced in the mitochondrial matrix is exported to the cytosol. And this uses the glutamate aspartate antiporter. Then this aspartate transfers its amino group to alpha-ketoglutarate. Alpha-ketoglutarate is converted into glutamate and aspartate is converted into oxaloacetate. It is catalyzed by the cytosolic aspartate amino transferase. Here also in mitochondrial matrix the concentration of oxaloacetate is more, so aspartate amino transferase catalyzes the reaction from oxaloacetate to aspartate, and in the cytosol the concentration of aspartate is more, so here this enzyme catalyzes the reaction from aspartate to oxaloacetate. Export of the aspartate from matrix to cytosol is coupled with the import of the certain molecule from cytosol to the matrix, and this molecule is glutamate. That means aspartate is exported from matrix to cytosol and glutamate is imported by the mitochondrial matrix from the cytosol. If you like this video, please hit the like button, share it and subscribe my channel. Thank you. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.